Welcome to Chapter 3, and in this particular chapter, we're going to be exploring more and more into the cell structures and things of that nature. So when I'm going through these slides, it's more commenting on them. I'm hoping that you look at the PowerPoint slides beforehand. So with the cytoplasm of a cell, there are many things that can be dissolved in the cytoplasm, which is the fluid portion of the cell. Obviously, organelles are in the cell, but there are also some inclusions, and the inclusions are vacuoles that might be able to store glucose or glycogen, store fat, and things of that nature. So it's a kind of a matrix in which to suspend all of these things. And when you are talking about organelles, they fall into two categories. Those organelles that have membranes, and the importance of membranes is the fact that they have a phospholipid bilayer, and then so they are membranous organelles. Non-membranous organelles means that they do not have a phospholipid bilayer. So for the membranous, most of the organelles that we think of as mitochondria or perioxysomes or lysosomes or endoplasmic reticulum or Golgi, the nucleus, they are all encased in a phospholipid bilayer membrane. Non-membranous means they don't have that, and that would be cytoskeleton, which are just protein rods, centrioles, which are also protein rods, ribosomes, which are a collection of RNA and protein. So the types of organelles will really depend on whether they have a membrane or not. So one of the most important organelles in a cell is the ATP energy generating mitochondria. They have two membranes, the outer membrane and then the inner membrane. And the inner membrane is the part that is folded to give you more surface area. Because remember, that is where you're going to be generating 38 ATP, and that allows so much surface area in order to accomplish that feat. So when you do cell respiration, that's where the oxygen and the hydrogen electrons are going to go down the electron transport chain. You also will have in the middle of the mitochondria the Krebs cycle. So in fact, the only thing in aerobic respiration that is outside of the mitochondria is the first stage, which is glycolysis. They are considered to be ancient versions of a very small aerobic bacteria that was able to coexist with a larger bacteria. Then one of the evidences for that is the fact that they contain their own DNA. The DNA in the mitochondria codes for genes about the function of mitochondria. The genes in the DNA in the nucleus is all about how to control everything else. Okay, so this is an example. This is a lovely picture of the double membrane and where you find ribosomes, what they look like under the scanning electron micrographs and things of that nature. Now, ribosomes. Ribosomes are two subunits, a large and a small subunit, and they are made out of a complex of ribosomal RNA and protein. Their real function is to hold on to a messenger RNA in the cytoplasm and help read it properly. So I always think about messenger RNA like a sentence. So it's going to have a beginning, middle, and end. And to read the sentence, ribosomes are almost like the bouncing ball, so that the cell actually knows that the ribosomes understand the beginning of the sentence, the middle of the sentence, the end of the sentence, in synthesizing this protein. They can be either free in the cytoplasm or they can be bound to the endoplasmic reticulum. If that's the case, then that is called the rough ER, the rough endoplasmic reticulum, and the ribosomes apparently make it look like it's rough. So the endoplasmic reticulum, it says it's interconnected tubes and parallel membranes, blah, 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 blah. But what the flow of information is in a cell that's trying to make a protein is that you have the blueprint in the DNA. You make a copy of the blueprint in messenger RNA. That leaves the nucleus, and it immediately is now going to be associated with the ribosomes on the endoplasmic reticulum, which is like a very, very long tube. As the synthesis of the protein occurs, the protein is going to be deposited in the tube and then get transported to a particular place in the cell where it's needed. That tube-like structure is the endoplasmic reticulum. It really makes the transport of that new protein be very efficient, very fast, and so the cell doesn't have to wait for the protein to diffuse across and somehow reach its destination. There are two types. If their ER is studded with ribosomes, it's called rough, and if it's not, it's called smooth. So I think this is perhaps the most important sentence in this particular PowerPoint, that the proteins that are being made from the messenger RNA and directed by the ribosomes are going to enter that endoplasmic reticulum, and it will eventually accumulate at the end. Now, at the end, the protein could be released to the cytoplasm, or if you want it packaged, then, and why would you want to package? And the Golgi apparatus is what's going to package is. You might want to have a large number of this particular type of protein. So as it's being made individually, then you can collect a fair amount of them in the Golgi apparatus, which would either be able to import or export large amounts. Smooth ER. Smooth ER means that it doesn't have ribosomes associated with it, so it isn't about making and transporting proteins. It's about smooth ER is about having enzymes that can assist the cell in metabolizing lipids or fat, like cholesterol. And that's important because fats are a source of energy, and you would certainly, as a cell, want to be able to store fat in places that then on a rainy day you'd be able to tap into that fat and get that energy. So the smooth ER is all about being able to enzymatically, metabolically assist in storing the fat, utilizing the fat. And fats are very, very rich in calories and can enter metabolic pathways and make ATP. So this is showing you the smooth and rough ER. Wherever you see little red dots, those are the ribosomes, and over here there are no red dots. So this is the smooth ER, and then this is the rough ER, and you can see that it's very much, both of them, associated with a nucleus, so that any information needing to go in or out to these endoplasmic reticulums, it doesn't take a lot of time transport. It's right there. The Golgi, named after Dr. Golgi, and the name stuck. So sometimes it's called Golgi body, sometimes it's called Golgi apparatus. And the whole idea of that is that it's a separate organelle that can accept vacuoles loaded with proteins from the endoplasmic reticulum and store them, process them, package them for export or import. And that's really the importance of this particular organelle. And the trans phase, cis phase, you don't have to get into. That's actually more information than you need to take the Moodle quiz. The other thing is that they mention lysosomes right here. Lysosomes are bags of degradative digestive enzymes. They are recycle centers of the cell. Cell organelles age, and when they age, the lysosome can engulf them and break them down to their component parts, which can be recycled and reused. And this is showing how you could transport proteins from the rough ER. You see the little transport vesicles, and then they merge with the Golgi. And then you can concentrate them, process them, package them for export or import. Perioxysomes. So these organelles are about trying to neutralize free radicals. Free radicals sometimes are made by different metabolic pathways, and they are highly reactive and very destructive. And I don't need you to know that oxidases convert hydrogen peroxide or catalases convert hydrogen peroxide to water and oxygen, but just that these types of free radicals are fairly common as side reactions, but they're very harmful to the cell, so that you need an organelle that would be able to process these and to make them harmless, and that's the job of the perioxysomes. And lysosomes, they're all about recycling, and they are able then to degrade any aging organelle and be able then to release those building blocks to synthesize something else. Okay, endomembrane system. The endomembrane system, and besides whatever is on the slide, what that is all about is the fact that we tend to talk about organelles as though they are static beings inside of cells that don't ever change or don't ever morph themselves into something else. And the endomembrane system is an attempt to talk about the fluidity of the membranes. Membranes, you can borrow from Peter to pay Paul. And what I mean by that is if you have a plasma membrane and you might need a little bit more of a piece of the phospholipid bilayer to make the nucleus larger, you actually can have the membrane from the outside of the cell go into the cell, fuse with the nuclear membrane. The nuclear membrane might need to pinch off a little bit of its phospholipid bilayer for the endoplasmic reticulum, and then it fuses and the endoplasmic reticulum can grow. Maybe you have to make a few more vacuoles for the Golgi body, and vacuoles are made up of phospholipid bilayer, and you would be able then to fuse that with the Golgi. It's a very dynamic system because all of those membrane enclosed organelles are made up of phospholipid bilayers. So you could have a phospholipid bilayer at 8 o'clock in the morning be part of the plasma membrane. At noontime, it could be part of the nucleus. And then maybe by 3 o'clock, it could be part of the lysosomes. The thing about the organelles is really the exception to the rule, and the exception to the rule is the mitochondria. The mitochondria has its own DNA. It has its own way of synthesizing its own phospholipid bilayer. The phospholipid bilayers of the mitochondria do not fuse with anybody. They are not part of the endomembrane system because they have their own DNA, and they are just about regulating the mitochondria, including their phospholipid bilayers. So when you look at this laundry list as far as what is in the endomembrane system, this is not a totally complete list, because I would start with the plasma membrane, nuclear membrane, endoplasmic reticulum, Golgi apparatus, secretory vesicles, lysosomes, And all of those phospholipid bilayer enclosed organelles can move their phospholipid, not all of it, but it could be, a length of it that is needed by another organelle, and that is what will happen within the cell. So it's a fluid, it's dynamic, it's ever changing. If the cell needs more phospholipid bilayer in the endoplasmic reticulum, then it will borrow from Peter to pay Paul. It will take it from whatever plasma membrane is nearby, and so it goes. And this is an example of the endomembrane system. They should not have mitochondria here. I would have taken them out of the diagram. But here you have smooth ER, nucleus, nuclear envelope, rough ER, Golgi, transport vesicles, lysosome, plasma membrane, secretory vessels. Everything but the mitochondria is in the endomembrane system. Cytoskeleton. You have an endoskeleton. That gives you the shape of your body. Every cell in your body has an internal skeleton as well. It's called a cytoskeleton because it's the skeleton of the cell. And if you have columnar epithelial cells, they are columnar because of their cytoskeleton. If you have squamous epithelial cells, they are really flat because of their cytoskeleton, etc., etc. So your skeleton is made up of a composite of calcium phosphate salts and collagen, which is a protein. And internal cell skeletons are also made out of proteins. And not so much mineralized salts, but definitely proteins. And the proteins come in 3 flavors, going from small to large: microfilaments, intermediate filaments, and microtubules. Microfilaments are the smallest. They're just 2 strands of protein twisted around each other, and they're great in assisting in cell motility. And then next, and that's just showing you the 2 strands. They're actually made up of actin proteins. Actin is a contractile protein, which is why they assist in motility. Now, you're going to say motility, the cells don't move. And the cells don't move. Motility means the movement of molecules in the cell. So that type of motility. Intermediate filaments, I always think about a really thick rope as an example of an intermediate filament. It's many, many fibers that are twisted on each other. So it's fairly massive, and it's going to help anchor different points of the cell. And here you are, the rope-like intermediate filament. The microtubules, they are amazing. They are hollow tubes. So you're going to say, hollow tubes, how do you get strength from hollow tubes? How do you get, because really the shape of a cell is because of its microtubules and the arrangement of the microtubules. But if you take a piece of paper and you make it into a hollow tube, and you take a book, it would actually, if you put the tube vertically on a table and then put something heavier than that piece of paper tube on top of it, it would be able to hold a fair amount of weight. And same thing. And of course, if you're making a hollow tube, it's easy to make. It doesn't take a lot of time. It still does a lot of structural integrity of the cell and the basic shape of the cell. The other thing about microtubules that I'm not going to get into right now is it can also be involved in cell division. It can be involved in cells that, like sperm, that have a flagella that needs to whisk that sperm up the female reproductive tract. So you can find microtubules in a lot of different, let's say, a lot of different situations in the cell. But the part that I'm going to emphasize, and that will be on the Moodle quiz, is that it is involved in maintaining and defining the shape of a cell. And there we go. Here's our little hollow tube and microtubules. The nucleus. I think we all understand that the nucleus is going to house the chromosomes. It's going to house the DNA. But in addition, it's also going to house a little element inside called the nucleolus. And that is right here, this little blue blob. And that is where you can synthesize ribosomal RNA and assemble the proteins in the RNA into the subunits of the ribosome. This is going to occur in the nucleolus, which is in the nucleus. And then the ribosomes are able to leave and go out and stud the endoplasmic reticulum through these pores, which are located in the nuclear membrane. The kind of, what would you say, purple dark staining area are the chromatin, which is unwound chromosomes, or DNA. And that's where the genes are, which then can be copied. The blueprint in the genes can be copied, and then leaving the nuclear pore, the messenger RNA can go out into the cytoplasm to be translated, and a protein can be synthesized. So this is the nuclear envelope, double membrane, has pores in it that allow movement of small molecules in and out. And the nucleolus, or nucleoli, because nuclei usually have more than one, and it is going to be involved in RNA, ribosomal RNA synthesis and ribosome subunit assembly. Chromatin, the unwound, and I say unwound because between cell divisions, you don't have chromosomes as we think of them. You don't see 46 chromosomes when you look at a nucleus that has finished mitosis. Instead, it seems like you just see these strands of very thin filaments inside the nucleus, and they are called chromatin. The DNA is still partially wound around what are called histone proteins, and the fundamental unit of the chromatin, which is the DNA protein RNA complex, are called nucleosomes. And when we do mitosis, which we will talk about later, then prior to mitosis, a couple of hours beforehand, and especially at the very beginning of mitosis prophase, they will start to condense and they will pack very, very closely. And then that's when you're going to visibly see the chromosomes as we think of them. And that way, the cell would be able to replicate them, and during that process of mitosis, 46 will go in one cell, 46 will go into the other cell, and then they will unwind again and to form that loose spaghetti-like strands of chromatin. So this is showing you what will happen right before mitosis, and you're going to be taking these strands and condensing them, concentrating them until they make this chromosome-like structure. And that's it. So I will see you soon for the next unit.